So what actually was this workshop that happened last week? So no offense to all the other presenters at this taster day, but I think it's the most important session that you could go to, because it's the idea of the workshop is to explain some of the issues and problems we've got currently in science, including in social science. So I kind of feel like it's all well and good learning these sophisticated statistical techniques and these methods, but unless you put those methods to good use, you're not going to be doing science and research a favor. So it's kind of like deciding how you're going to use your statistical and your methodological, your knowledge. Are you going to use it for good or for bad? And so the idea of the workshop that I ran last week is really to try to encourage you to use it for good, use your knowledge and your skills for good, and explain how to avoid transgressing into the dark side, I suppose. So what am I really going on about? So this was one of my favorite articles that was published about 15 years ago. I love this title, Why Most Published Research Findings Are False. And in this paper, John Ioannidis was essentially saying a lot of published research in the scientific literature has to be false or can't really be true. And he did a bit of statistical wizardry to try and demonstrate this. But it was really this paper that kind of highlighted a lot of issues in the scientific literature, which has gone on to be called the reproducibility crisis. So what do we mean by the reproducibility crisis? It's essentially where lots of our previous studies, our previous research that's been published, has been shown that we can't reproduce those results. And it's kind of like a fundamental cornerstone of science, really, is that you should be able to reproduce the results of any study or experiment if we're going to believe them and think that they're valid. But unfortunately, lots of our studies in lots of different disciplines have been shown that we can't reproduce them, which raises a lot of questions about how accurate and truthful much of the research in previous literature actually is. And so this is an issue that goes across all scientific disciplines, really, well, most of them anyway. For example, in psychology, they found when they looked at 100 different sort of quite significant studies that had been published in the past, almost 2/3 of those couldn't actually be reproduced. And we get a similar trend in other areas of research, so medical research, or lack of reproducibility in areas of medical research, economics as well, a degree of lack of reproducibility, and other fields as well. And so why, you might be asking, why have we got these issues in science, these inability to reproduce previous findings? And I quite like this way of trying to explain some of the issues, and this is really what the workshop last week was all about, was trying to explain these issues and how we can overcome them. And so I will briefly explain some of these issues, and I'm going to use this sort of metaphor of the 4 horsemen of the apocalypse, but actually it's the 4 horsemen of the reproducibility crisis. So it's not me that's coined this term, it's actually Dorothy Bishop, who's a well-respected and well-known person in the whole kind of field of meta-science and reproducibility. So very quickly, just to run through these 4 horsemen of the reproducibility crisis, these are sort of 4 issues that contribute to the problems we've got in science at the moment. So the first one is publication bias. This is essentially where most journals currently, they have a bias towards publishing positive results. So by positive, we mean where you've found the thing that you were looking for, I suppose, or you found a significant result. You've found that sort of hallowed p-value below 0.05. This is problematic because journals tend to favour publishing positive results. It means there's a sort of, often we end up with this file drawer of unpublished negative or null results. So it kind of skews the literature. It makes it appear as though a certain topic may be accurate when it's not. But the other problem with publication bias is that researchers tend to want to get a significant or positive result, because getting publications is kind of like our lifeblood. It's the lifeblood of academics. So we want to publish papers to improve our careers, and so therefore it encourages us to actually try to get a positive or significant result in our analysis. And this might lead us to something called questionable research practices, where we aren't perhaps being as objective and rational as we should be. And one of the things that might happen if we're trying to seek out these positive, significant results is something called p-hacking. And I really like this quote. So if you torture the data for long enough, it will confess. And so p-hacking is essentially the idea that you can selectively manipulate and analyze and report your data in a way that you can eventually get it to show basically whatever you want. And so that's obviously not a particularly objective way of approaching your data analysis. And a really good example of this emerged in the psychological literature, where a paper was published in a really well-respected psychology journal that was essentially presenting evidence for extrasensory perception. So kind of going against all known laws of physics. So extrasensory perception, this idea where you can kind of see into another room, or you can predict the future. And this paper managed to get published because it appeared to be sort of well-run studies with appropriate kind of scientific methods. But what it really was an example of p-hacking, where the author had been able to selectively analyze their data and kind of only select significant results and report those to make it appear as though there's a reasonable evidence base behind this sort of completely unrealistic phenomenon. And one of the reasons we're able to p-hack data, and this goes for all researchers really who are handling data, is we have lots of degrees of freedom in how we analyze our data and what we do with it. So for example, there's lots of choices we can make about the variables we explore, what data we gather, how we code our data, how we analyze it, for example, how we might interpret it. So all these different choices, and with all these different choices, we've got lots of different ways we can analyze our data. And so usually we can find one of those ways that gives us a sort of significant and positive and publishable result. And this was also demonstrated in another paper where the authors were deliberately p-hacking their data to demonstrate that you can essentially show almost anything depending on how you analyze your data because of the amount of choices we've got. And so in this paper, they actually showed that through the sort of flexibility of how they analyze their data, that by listening to a certain song actually makes you physically younger, which is completely ridiculous, but they kind of showed it through their the way they were able to analyze their data. And then another one of the problems or issues that has led to this reproducibility crisis that I mentioned is something called harking. And this is where we hypothesize, we form hypotheses after we've known our results. So it's essentially sort of seeing what our results tell us and then claiming that we predicted that result in advance, and that's problematic. Another issue, which actually got alluded to a little bit in the sort of earlier conversations today about sample sizes and power analysis, is that in many of our studies, particularly in kind of more fields that use more sort of experimental methods, with sort of experiments or intervention studies, often our samples are too small. So the number of observations we've got are just too small to be reliable. And so we can see this through this concept of power, which is a statistical power, which is essentially the likelihood that you will be able to find an effect within your data if what is truly there, if it is truly reflected in the wider population. So usually we want quite a high power because we want to be sure that we find an effect in our data if one is truly present. So we usually aim for this 80% sort of threshold of power. And most studies in lots of different disciplines tend to fall well below this kind of acceptable level of power. And you can kind of imagine what might happen if we're using small samples. If we use a small sample of data and our studies had low power, so if it's had low power, that means we've got less chance of actually finding an effect, even when an effect might truly be there. So if we've done a study that had low power, we end up not finding any evidence for our hypothesis because we had low power in the first place. What might this lead us to do, perhaps, given the fact that we know that journals like to publish positive significant results, what might it do? So it's kind of inherent within our kind of publishing system that it kind of leads us towards these questionable research practices where we might end up p-hacking data, for example. We might try to torture our data in order to eventually find something that we can publish. So that's a very sort of quick summary of some of the issues that we've got in science at the moment. So p-hacking, harking, the sample sizes we're using and statistical power of our studies, and the wider issue of publication bias within journals. And so when I explain all this, you might be sort of, perhaps it's slightly disheartening. You might be a bit wondering, you know, what have you done getting into this whole field of science and this whole sort of nest of questionable research practices. But the whole point of the workshop that was run last week was to actually hopefully give you some pointers and tasters and signpost you towards how and different ways in which you can do your research better and hopefully avoid some of these issues that I've just talked about in the last sort of 5-10 minutes. So very, very quickly, as I'm conscious of time, these were some of the things that we talked about in the workshop last week. So things like pre-registration, using open data and reproducible analysis that people outside our research team can go and have a look at and reproduce our results. Using preprints, for example, collaborating much more so we can pool our resources and increase our sample sizes. Obviously, using appropriate sample sizes, things like systematically reviewing our literature before we start on a study, so that we're not cherry picking evidence and we're getting a much more objective view of the past literature. Things like triangulating our methods and our data analyses, so we hone in on a certain finding, and replicating previous results as